So welcome to FlexBio. We're standing here in the main fermentation hall. I'm going to take you around FlexBio a little bit more in just a few minutes. First of all, what I want to talk about is what the bioprocessing scale-up centres do and split across, as I said earlier, over in Rapid Bio in Strathclyde University. What we tend to do there is focus more on the smaller scale processes, maybe looking to do what we call downscaling processes, to be able to look to do process improvement on them in almost like a microliter level before then scaling up. Over in FlexBio, generally what we do is we take what we call shape flask cultures and we scale them up to larger scale. And I'm going to show you in a little while what that means. But just so you can get your bearings, we're stood in the main fermentation hall at the minute. We're going to go and see what we call a preparation lab, where we make reagents and buffers that we do our fermentations with. We also do a lot of material manipulation when we've maybe harvested material, and we can manipulate that. In another smaller lab, we've got our smaller scale microbial culture lab, and we're going to show you some smaller vessels there before coming back into the main lab here, showing you our larger system and showing you what we do with the cultures, microbial cultures, once we've grown them up, and how we look to purify that material as we take it through downstream processing. So yeah, if you want to come with me, we're going to go downstairs and I'm going to show you what type of scale we normally start our processes, what customers and industry come to us with, and how we look to scale that up. So come with me. Cool. So thanks for joining me back here. So we're back down in our fermentation hall, and what I want to introduce to you first of all is the idea that a lot of our customers that come to us are industry, so companies wanting to perform scale-up studies. Some of them are academic groups that have done research, they may be working with a company and they're looking to scale up that idea. And quite often a lot of our cultures come in these flasks here. These are what we call shape flasks. This is just an empty shape flask with some media in. This is just LB media. But it's the idea, it's called a shape flask. Generally, when the bacteria or microorganisms are in there, we just shake them on an incubator. And this has got our bung in the top here. It's also got foil on the top, and that allows some air diffusion through the vial, through the vessel. But the important thing to get across to you at the moment is when this is shaking on a shaker, the amount of air that can get into the vessel, the amount of shaking, and so the amount of real oxygen that really can get into the cells for them to metabolize is actually very low. We've got no way really to see how much sort of carbohydrates, so that's the food that a lot of cells will eat, or protein. We've got no way really of measuring that, short of taking it off the shaker and measuring that. So what we need is a more reliable way to grow shape flask cultures. But a lot of the processes that come to us have only been grown in these very limited conditions, so limited for oxygen, limited for carbohydrates, and limited for other nutrients, as well as being limited for in terms of controlling the acidity or alkalinity of it, so the pH of the solution. So what we need is a larger solution than this, so we can start growing up, because obviously we can't, you know, a lot of manufacturing processes exist on scales such as, you know, up to 100,000 litre scale. So the idea you could have a 100,000 litre shaking flask is pretty nonsense. So what we have to do is we have to adapt how we grow the culture. So we have to do it in more a vessel, and we have to mechanically agitate the shells in a much better way. And the example I've got just here for you, this is one of our 2-litre gasket vessels. So the idea here is, yes, it's made out of glass. As we get bigger, they can get made out of stainless steel and other materials. But what we've got in here is, again, an enclosed vessel. But we've got what we call an impeller here. It looks a bit like a propeller, and that basically turns to agitate the culture. And at the bottom here, we've got a little L-shaped metal probe. Inside of that allows us to attach it to an air supply. So we can now put air directly into the culture, so that's good. We can stir now, and we can stir really fast, so that's good. We've also got another long metal pipe here, and what that does, it goes directly into the culture, and that allows us to do sampling. So now we can do samples in real time if we want, in terms of what's happening in the culture, how fast are the carbohydrates being used up. how fast. We can also monitor for other things. So, for example, we've got two types of probe. This is a pH probe. That's going to go into the culture, and that's going to help us maintain a stable pH. So maybe if we've got pH set point of pH 7, it can add acid and alkali as the pH changes in real time. And that will simply just go in straight through the top of the vessel. We've also got what we call a dissolved oxygen probe. So rather than just guessing, or in our shake flask culture, just stirring and hoping oxygen gets into the culture, we can not only inject air directly into the culture, we can also monitor that using a dissolved oxygen probe. And a dissolved oxygen probe is really a membrane on the end of an electrode inside, and that's got electrolyte in, and it's able to allow oxygen to transfer across the membrane if you look after it very well. And we can, again, insert that directly into our culture, and changes in oxygen in the solution can be directly fed back to the controller for air, so we can inject more air into the culture and less air if we're getting enough oxygen in there already. So what I want to do is just show you now the next step in it. So that one you just saw was a 2-litre vessel. I would like to show you here, this is our 30-litre vessel. So this is essentially very similar to the 2-litre vessel. In fact, one of the biggest jumps and one of the most technical challenges is actually going from this scale to that 2-litre scale, being able to adapt our culture, so many unknown things. We have to work very hard to scale it to a successful culture at 2-litre scale. Once we've got a good idea of how it grows at 2-litre scale, we can then have a lot of numbers from them, and we call them like tech transfer parameters. But we can really feed those parameters in, so we know stirring rate, we know airflow, we know how much pH control is needed. We've got a good idea now in terms of the feeding of the fermenter as well. So if it needs more carbohydrate or more other nutrients, we can add them in while it's running. And so we can be controlling this remotely as well. We can be doing this at 2 a.m. in the morning if you like. We try not to, but sometimes cells grow at very unusual rates, and sometimes don't grow as expected. So being able to control them from home is very, very useful for us. So yeah, this is a 30-litre vessel. But typically, when we say 30 litres, we actually talk as well about the working volume. So the working volume is generally up to about here, allowing a good space in the top, which we call the head space, and that allows the culture, when it's being mixed, to build up any foam and not cause any foam to go any further up the fermenter. And there's various strategies to control that. But I've skipped over really some of the scale-up steps in the idea, just to give you a bit of an idea of what we do in FlexBio. I'm now going to take you into some of the smaller labs to show you how we do some of those medium-sized steps. And I think it's important to mention at this point that while this is a 30-litre vessel, it's made of stainless steel, we can sterilize it right here using steam. These go up to huge sizes, up to 1,000, 10,000, 100,000, and even higher scales than that. So this is really a very small vessel compared to what manufacturing scale will be. But this is the first technical challenge on a journey from an idea of growing and engineering a cell, maybe using genetic manipulation to change an E. coli cell or a yeast cell to produce some valuable product. We're actually able to take those genetically engineered cells and grow them up very slowly up to this scale. And once we can do that, it's really then about just working through the engineering maths to really work out how you can scale up much larger than that. But as we specialize more in the smaller scales, I'm really keen to show you about some of the medium steps. I'm going to take you through some of the lab through there. We'll come through to one of our smaller laboratories now. We've talked about our shake flask. We introduced the idea of the 2-litre vessel. What I'm stood next to now is actually more of those 2-litre vessels, but these are actually, when we have the other one that's in the other lab at the minute, we actually have 4 of these. So what that means is, like our 30-litre culture, we can control the stirring speed, the airflow, the pH, we can sample, and we can control feeding in carbohydrates. We can actually do that on 4 vessels at one time. So say if you're wanting to look at different pHs, different airflows, or different feeding rates. We can actually do that completely independently for the 4 different vessels, and we call that parallel fermentation. Well, what that allows us to do is do 4 completely unique cultures, even grow up different cells side by side, because we tend to sterilize these and we inoculate, so we add in the cells. So that shake flask culture you saw earlier, we use a culture of that size to inject into our culture here, and that process is called inoculation. And when you're starting to move from shake flask, which you would typically do in a biosafety cabinet, to move into this scale, you tend to have to sterilize these vessels outside in a large autoclave. And then we tend to take that shake flask material, that smaller volume, maybe 100 mils, that type of volume, and we inject that into the vessels in a sterile manner to not introduce any contamination in there. But really, this is a very flexible system. So again, we can be controlling this 24 hours a day around particular set points that we want to control that. And it allows us to see in literally second by second timescales small slight changes in the vessels, because while a lot of bacteria may grow and have particular requirements for acid and alkali, there can be certain points in time where they start to run out of particular food sources, where they switch over and start doing different types of metabolism. So that allows us to then see where it starts to consume acid and alkali, and that gives us a real good sense of where the bacteria is maybe limited in terms of how it's growing as a culture. So these 2-litre system, this is a gas skip system made by Eppendorf, are a really, really good workhorse for us to do in process development, and particularly that first challenging step from shake flask to a stirred reactor. In FlexBio, we actually have an 8-pod system of this, so we have 8 of these vessels, and you can imagine how challenging it is with all the different air lines. So you've air in, air out, you've got feed lines, acid and alkali lines, you've got to be able to connect your pH and dissolved oxygen probes. So there are a huge number of wires and pipes here, and even further more that are not on here currently. But really, we have to be very, very careful and work to a very high standard to be able to control and make sure we've connected everything with that. And I'm going to show you in a little while some of the documentation that we use. So while I mentioned that we don't work to sort of GLP and GMP, a lot of the way we work is very much in the spirit of that. So particularly working very strictly to standard operating procedures, SOPs, and working, filling in a lot of logbooks to make sure, of course, that every single one of these lines is connected, and we've run tests on the vessels before and after to make sure they're running exactly as we expect them to run as well. So we're going to go through now, and I'm going to take you through, show you another type of technology that we use for doing scale up. So now what we're going to do is we're going to show you a different type of culture. At the moment we've got a client in the lab, and they're requiring to use a slightly different type of culture. So you've seen the vessels that we've had previously are more traditional stirred vessels, so called like stirred tank fermentation. What we've got here is a rocking system, so people call it wave bags. This one's again by the company Applebomb. This is an Appleflex system, and what this essentially does is it's a very, very cells that are very fragile, perhaps, maybe bacteria, for example, they don't mind being in a stirred reactor. E. coli, yeast, other organisms. You can stir them a lot. Sometimes you can't stir them enough. You just can't get enough air in. Whereas in a system like this, so for example, used with mammalian cells, stem cells, plant cells, the idea that the gentle rocking motion and you've still got air inlets as well, so you can see there's an air inlet here. It allows a very gentle introduction of air, and the rocking system is very, very much more gentle than the impellers you saw on the stirred harder systems. So again, lots of similarities to what you've seen previously. We've got air intakes, we'll have an air outlet. We've got dissolved oxygen probe in the top here. We've got a temperature probe. We've also got a pH probe, so we can control addition of acid and base where we need to. And we can also control the rocking, as well. So this is rocking at a particular speed, and the client will be interested to look at particular parameters. I don't know actually in particular which parameters they're looking at, so I can talk in general terms. We always have to be very careful on confidentiality. So on this, they may be looking at different rocking speeds, or they might be looking at different pH set points, or different airflow rates, different temperatures, and this system allows us to do that. But instead of doing them in the stirred reactor, it allows us to do it on a very gentle wave rocking system as well. And this is all connected to a fermentation controller unit. So this is what we call an easy controller unit, again connected with the Aquacon system. And this allows us to actually document exactly what the trend lines are like on the thing. So it allows us to show graphs on there. We've got a computer around the corner as well, so that's recording in real time. And again, we can remote access into the PC. We can set up a webcam here as well, so we can watch the culture at night if we so choose to wish. And we can access the software, and if we need to make any changes to the fermentation based on perhaps physical numbers that are coming up on the screen, or maybe visual occurrences within the culture, we're able to do that remotely from home. So this is an example of a wave bag system. It allows us to be very, very gentle with the cells, but still encourage them to grow. So where we're going to go next is we're going to go back to our main fermentation hall for the final part of the tour. There you're going to see our larger fermentation system, and then we're going to start talking a little bit more about what happens when you grow the cultures. What happens to them then? In some processes, they want the cells, that's all they want. But in some processes, so for example, high value pharmaceuticals, so antibodies and antibiotics and these high value pharmaceuticals, you sometimes have very, very in-depth downstream processing. So we're going to look at what happens downstream of growing cultures next through in our main fermentation hall. So I invite you to come through with me back to that. Hi, so we're back in the fermentation hall, back with our 30-litre sterilisation in place vessel. This vessel, as we've mentioned earlier, is a stainless steel vessel. It's actually what's called a double jacketed vessel. So it's actually got two layers. It's a bit like a thermos flask, but instead of in the thermos flask, it's a vacuum. In the sort of outer layer, the outer compartment, we're able to put steam in there, and that really allows us to surround the vessel completely in steam, and that helps us get it up to roughly 120 degrees to be able to do a sterilisation. When we then want to cool that, we can then use cooling water. So that cooling water comes from another building, and that's piped in, and then the warm water that's used to cool is then piped back to another building. And that allows us to do a really good sterilisation. So we don't need to autoclave 80 litres of media outside the vessel. We can actually do that inside the vessel. And generally, the largest style fermentation systems, as you start getting much larger than this, steam will be a really critical part of enabling you to autoclave and sterilise your media beforehand, before you then bring your smaller culture in to inoculate. So like you saw previously, our shaking flask culture is used to inoculate our 2-litre vessel, and a volume smaller than our 2-litre vessel will be used to inoculate this size of vessel. Likewise, this size of vessel might be used to inoculate a 300-litre vessel, and sometime in the future at FlexBio, we've got aspirations and plans in place to get a similar size to a 300-litre vessel, which will allow us to do really powerful upscaling work in microbial cells, really quite unique to anywhere offering in Scotland at the moment. So at the moment, we've got 30-litre, hopefully shortly to increase to 100 and then beyond. But what I'd like to do just now is explain what happens when the culture's finished. So there's lots of different things that might happen to a culture when it's finished, depending on what the final product is, how pure that product needs to be. And it's important to explain at the moment that if we were to harvest the product, so when we say harvest, we mean empty out the cells and media from the vessel into basically a capture vessel, so almost like a large bucket. What we've got to have in there is we've got the media it was growing in, which also contains a lot of waste products, but also we've got the cells that were grown in it. And it's important to realise that sometimes the cells are the product, sometimes the product is inside the cell, sometimes the product is grown on the outside of the cell, on the membrane.
but sometimes the product has been produced by the cell into the media. So depending on where the product is you're interested in really determines what happens next. And as I've mentioned previously in the talk, downstream of the upstream process is what we call downstream processing. So I'm going to take you through some techniques now that typically help us to isolate the product depending on where it is in the vessel. So thanks very much for joining me on our virtual tour of FlexBio. Thanks also to my assistant Naomi, who's been helping me very ably on the camera. Hopefully you've got a good idea as to what we do in FlexBio and some of the processes and procedures we need to follow to do a microbial scaler. We've got lots of plans in place in future to do much larger work. We've got some really exciting projects in the pipeline to work with different industry companies, and so I look forward to sharing further updates with you in the future.